Each tape in this series demonstrates a complete dissection of a particular specimen. The tapes can be used as pre-lab dissection demonstrations or as alternatives to dissection. Each tape focuses on all the major systems of a selected species and illustrates proper dissection techniques. A printed script is included with this video. If your VCR has a real-time counter that displays hours, minutes, and seconds, you can utilize the numbered frame references listed in the script at the beginning of each major section. Set the counter on your VCR to 0 now. We will now proceed with the dissection of the fetal pig.
Pigs belong to the phylum Chordata, class Mammalia, order Artiodactyla, genus sus, species Scrofa. The fetal pig is an excellent representative of its class of mammals because it illustrates the basic anatomy of the adult mammal while providing information about certain features and structures only present in the fetal body. In this specimen, red latex has been injected into the arterial system through one of the umbilical arteries. Blue latex has been injected into the venous system through the femoral vein near the ankle. The body consists of the head, neck, trunk, and tail. The cut umbilical cord is seen on the ventral portion of the abdomen. During fetal life, the cord, including its contained blood vessels, connects the fetus to the placenta located on the uterine wall of the female. On the head are the external ears, the eyes, with their upper and lower lids, and a relatively large mouth. The tongue is seen protruding from the mouth. The external nares, or nostrils, are located just dorsal to the mouth. The external opening for solid wastes, the anus, is located directly under the tail. In the forelimb, you can recognize the digits, the wrist, the elbow, and the shoulder. In the hind limb, the digits, the ankle, the knee, and hip. Now determine the sex of the specimen. The presence of a swelling called the scrotal sac at the caudal end of the body, as well as the presence of the urogenital opening just caudal to the umbilical cord, indicates that the specimen is a male. It is also possible to feel a long, muscular, tubular structure lying under the skin proceeding from the urogenital opening. This is the penis. In the female, the urogenital opening is located immediately ventral to the anal opening. Projecting from the urogenital aperture is a small, fleshy genital papilla. Both the male and the female have 2 rows of mammary papillae, or teats, on the abdomen, with 5 to 7 nipples in each row. Place the specimen dorsal side down in the dissecting pan. Tie strings around the lower ends of the forelimbs and the hind limbs on one side of the body. Run the strings under the pan to the opposite side and tie them to the forelimbs and hind limbs on that side. Make an incision beginning at the small hairy papilla on the upper part of the throat. Continue the incision to the umbilical cord. Cut around both sides of the cord. If the specimen is female, which is the case here, direct the 2 cuts into one and continue back between the hind legs. If the specimen is a male, proceed down to the umbilical cord. But instead of bringing the cuts together behind the cord, continue the two incisions separately between the hind legs. This is done to avoid cutting the penis, which lies under the skin along the midventral line. Next, deepen the incision to cut the membrane called the peritoneum. Now the body cavity is open. If the cavity is filled with dark fluid, flush it out with water. Separate the diaphragm from the body wall by cutting the edges of the diaphragm where it connects with the body wall. In order to make lateral flaps, cut into and through the body wall just caudal to the forelegs and just anterior to the hind legs. Pin the flaps down, out of the way. Look for a dark, tubular structure leading from the umbilical cord into the liver. This is the umbilical vein. Tie a small piece of string around the vein in two places. Cut through the vein between the strings. The umbilical cord and its attached strip of flesh can now be pulled back. The two major body cavities are the pleural or thoracic and the peritoneal or abdominal. In the following section, make only general observations, concentrating on size, color, and the relative positions of the organs. The diaphragm is a musculotendinous sheet separating the pleural cavity, which contains the heart and the lungs, from the caudal peritoneal cavity containing the visceral organs. The thymus glands are large, elongated masses extending from the throat region down over the top portion of the heart. The thyroid gland is an oval gland located near the base of the larynx. Now observe the heart and lungs. The liver is a large lobe structure lying just below the diaphragm. The stomach is a large sac located in the left side of the abdominal cavity and is almost completely covered by the liver. The spleen is the elongated organ attached by the greater omentum to the bottom of the stomach. The small intestine fills most of the abdominal space. The large intestine is a compact, darker mass of coils. The cecum is a small pouch-like sac at the beginning of the large intestine. And the urinary bladder is the large sac found between the two umbilical arteries. The digestive system of the fetal pig consists of the alimentary canal, including the mouth, the esophagus, the stomach, the small intestine, large intestine, and associated glands including the salivary, gastric, intestinal, liver, and pancreas. There are 3 pairs of salivary glands in the pig. By removing the skin and muscle layer from one side of the face and neck, expose a large triangular gland. This is the parotid gland. Note that it is composed of many small nodules. The mandibular gland is comparatively large and somewhat lobed. The sublingual gland is behind the mandibular. The secretions from these salivary glands maintain the mucous membranes of the mouth and pharynx in a moist condition and lubricate food for ease in swallowing. The glands also produce enzymes that initiate carbohydrate digestion. With the scalpel and bone shears, cut back through the corners of the mouth. Continue the cut until the lower jaw can be dropped, allowing the interior of the mouth to be observed. Examine the oral cavity. First, look closely at the surface of the tongue and observe the numerous papillae that contain the taste buds. Teeth may be evident in the fetal pig, depending upon its age. The hard palate is composed of bone and is covered with a mucous membrane. The soft palate is the caudal continuation of the mucous membrane that covers the hard palate. The pharynx is the crossing of the air and food passages. The opening into the trachea or windpipe is called the glottis. Note the presence of a small white tab of cartilage. This is the epiglottis. It prevents food from getting into the trachea. The esophagus is the muscular tube leading from the pharynx to the stomach. Notice its close association with the trachea. Examine the stomach. The long convex border is called the greater curvature. Concave and opposite is the lesser curvature. The strong muscular ring at the border of the stomach and the duodenum is the pyloric sphincter. It serves to close off the lumen of the stomach from the duodenum, except when food is passing through. The duodenum is the first portion of the small intestine. It leads away from the pyloric end of the stomach. The pancreas lies between the stomach and the duodenum. The jejunum and the ileum are the two regions of the small intestine extending as far as the large intestine. In this examination, they cannot be differentiated from one another. Remove a small segment of the intestine, about 1.3 centimeters. Open it longitudinally. Flatten it out and examine it. Notice the velvety appearance of the internal lining. This lining is composed of numerous finger-like projections called villi. As observed earlier, the cecum is the first part of the large intestine. The compact coiled mass following the cecum is called the colon. The caudal part of the large intestine, continuing from the colon, is the rectum. It terminates in the anal opening. The liver, the large gland, is divided into four main lobes. The gallbladder acts as a storage sac for bile secreted by the liver. Trace the common bile duct from the liver to the duodenum. A pair of kidneys is found against the dorsal body wall. They're covered by a smooth, shiny membrane called the peritoneum. This must be removed to see the kidneys clearly. Emerging from the kidneys is the ureter, which empties urine into the urinary bladder. The bladder is an elongated sac lying between the two large blood vessels known as the umbilical arteries. The bladder continues into the umbilical cord. Proceeding from the urinary bladder is the urethra. Expose the urethra by cutting carefully through the cartilage between the hind limbs. Now, follow the urethra to its union with the vagina in the female. If the specimen is a male, follow the urethra to its continuation with the penis. Next, look on the medial face of the kidney for the adrenal gland, which is part of the endocrine system. Hormones secreted by the adrenal glands go directly into the bloodstream and help to control a number of bodily functions such as heartbeat, regulation of blood sugar level, and development of certain secondary sexual characteristics. Remove one of the kidneys. Make a sagittal section through the kidney and study its cut surface. Observe the renal pelvis, the expanded portion of the ureter within the kidney. The central part of the kidney is called the medulla. Surrounding the medulla is the area known as the cortex. The ovaries are small bean-shaped bodies lying on the dorsal body wall just below the kidneys. Each ovary is suspended by a sheet of connecting tissue called the broad ligament. The small coiled tubes on the dorsal face of each ovary are called the uterine tubes. The uterine horn is the continuation of the uterine tubes. The muscular tube directly continuous with the body of the uterus is the vagina. Locate the scrotal sac. Slit the scrotal sac and pull out the dark-colored sac. Remove the sac to expose the testes. The gubernaculum is the tough white cord situated at the caudal end of the testes. The epididymis appears as a tightly coiled mass of tubules. The vas deferens proceeds from the tail of the epididymis and empties into the urethra. Together, the vas deferens, the spermatic artery and vein, and the spermatic nerve form the spermatic cord. The penis is the white tubular structure previously observed. The two large elongated glands lying between the urethra and the penis are the bulbourethral glands. The circulatory system consists of the heart, the arteries that carry blood away from the heart, the veins that carry blood to the heart, and the lymphatic vessels and nodes. This specimen has been injected with colored latex, the arteries in red and the veins in blue. Remove the thymus gland, the thyroid, and the muscles in the neck region. Now cut and remove the pericardial sac that surrounds the heart. Identify the heart's 4 chambers: the thin-walled right atrium and left atrium, the thick-walled right ventricle and left ventricle, the coronary artery and coronary vein lie in a diagonal groove between the ventricles. The large vessel leading from the right ventricle is the pulmonary artery, which in the adult pig carries blood to the lungs to be oxygenated. The large vessel leading out of the left ventricle is the aorta. It turns almost immediately caudally, continuing through the thoracic and abdominal cavity. The point at which it turns is called the arch of the aorta. From this arch, two major vessels arise. One of these is the brachiocephalic artery. It gives rise to the carotid trunk leading to the head and face, and the right subclavian artery leading to the foreleg. The other major branch arising from the arch is the left subclavian artery, leading to the left foreleg. Existing only in the fetal pig is a short connection between the pulmonary artery and the aorta called the ductus arteriosus. Because the fetal lungs are not functional, blood from the right ventricle is shunted into the aorta rather than flowing into the lungs. The aorta runs through the thoracic and abdominal cavities near the spinal column and branches out to renal arteries supplying the kidneys, lumbar arteries supplying the muscle of the dorsal abdominal wall, and the gonadal arteries, which are the ovarian in females and the spermatic in males. The external iliacs give rise to the femoral arteries which supply the hind limbs, and the internal iliacs that give rise to the umbilical arteries. Renal veins receive blood from the kidneys. Gonadal veins receive blood from the reproductive organs. In the male, these are the spermatic veins. In the female, the ovarian veins. Common iliac veins are the paired vessels forming the major caudal vessels. The external and internal iliac veins are the continuation of the common iliacs and receive blood from the femoral veins and the pelvis viscera. The umbilical vein is the vessel you tied pieces of string around when the body cavity was first opened. Veins cranial to the heart collect blood from the head, neck, cranial appendages, and thorax. They empty into the right atrium of the heart through the cranial vena cava. The superior vena cava is the large dark vessel that enters the right atrium. The external jugulars are a pair of veins carrying blood from the head and face. The internal jugulars drain blood from the brain. The vessels coming from the forelimbs are called the subclavians. They enter the brachiocephalic. The larynx is made of 4 cartilages, the largest being the thyroid cartilage. The trachea continues from the larynx to the lungs through its branches. The lungs consist of 4 lobes on the right and 2 lobes on the left. The 4 on the right are the apical lobe, the cardiac lobe, the intermediate lobe, and the diaphragmatic lobe. The 2 on the left are the anterior lobe and the diaphragmatic lobe. The lungs of the fetal pig are filled with water rather than air and are therefore more dense than the lungs of an adult. To expose the brain and the spinal cord, begin with a scalpel and make an incision around the upper portion of the cranium. Extend the incision along both sides of the spinal cord to the tail. Next, deepen the incision using scissors. Be careful not to cut or damage the spinal cord. Now peel back the skin with your fingers. Again using scissors, very carefully cut along the exposed portion of the skull. Now, using forceps, gently lift the cut section of skull. After removing the top of the skull, you'll find a membrane called the meninges. Remove it carefully. Now, identify the gross features of the brain. The cerebrum is divided into the right and left hemispheres by a longitudinal groove. A smaller mass caudal to the cerebrum is the cerebellum. The medulla oblongata is continuous with the spinal cord. A pair of olfactory bulbs is located on the ventral side of the cerebrum. The spinal cord is enlarged at the level of the forelegs, the cervical enlargement, and at the level of the hind legs, the lumbar enlargement. The spinal cord narrows at its caudal end to form a thin strand called the filum terminale. 33 pairs of spinal nerves are associated with the spinal cord. 8 of these are cervical. 14 are thoracic. 7 are lumbar. And 4 are sacrals. The union of a dorsal and ventral root forms each nerve. This concludes the dissection and observation of the fetal pig.
Many of the following items are featured in our dissection videos. We carry a wide variety of dissection specimens from invertebrates, including starfish, earthworms, mollusks, crayfish, and insects, to vertebrates, including fish, frogs, rats, fetal pigs, and cats. In many cases, you may choose from a selection of plain, single, or double-injected specimens. Sex-specific and pregnant varieties of some species are also available. We also offer specific organs for study, including hearts, eyes, kidneys, brains, and skeletal systems. Our selection of dissection supplies includes everything from disposable gloves to dissection containers and trays in a variety of sizes and configurations. We also carry a wide assortment of quality stainless steel instrument kits, from basic beginner's sets to larger advanced dissecting sets. Choose from hundreds of other dissection supplies in our catalog, including specimen bags, tongs, pins, dissection pads, syringes, forceps, and more. You'll also find our catalog filled with the dissection teaching aids the teachers request most. Our complete section includes the most popular lab manuals, charts, film strips, and videos. We also carry models, complete dissection kits, and a full line of microscopes to meet every classroom need.